Hereby I open this academic ceremony in which Magdalena Schellenberg Konopka will defend the academic thesis Critical Appraisal of Sports Genetics with an emphasis on endurance. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you. Dear Pro-Rector, supervisors, and members of the Corona, dear colleagues, friends and family, thank you all for being here today for the defense of my thesis. Over the next 15 minutes, I will provide a summary of the research I have conducted during my PhD. We all know that we have cells in our body, and inside each cell we have our DNA stored. And small segments of this DNA are known as genes. These genes have a special job. They code for proteins, which help our body to function. Our DNA is packaged into 46 chunks called chromosomes. And we inherit 2 copies of each chromosome, 1 from each of our parents. And because we have 2 copies of each chromosome, we also have 2 copies of each of our genes. When these copies differ from each other, we talk about genetic variants. If the difference is as small as a single nucleotide, we call it a SNP, or single nucleotide polymorphism. These small differences in our DNA can result in different traits that make us who we are. There are many traits, from your personality to the risk of developing diseases as breast cancer, and even sports-related traits are determined by genetic variations. So, we research genetics to find out which genetic variants are linked to a certain trait, which may increase an individual's susceptibility to that trait. In my research, I focused on sports genetics, which has the potential to optimize performance, to prevent injuries, and to contribute to the health and well-being of athletes. On this slide, you see a skier who won 7 Olympic medals. And in the 1990s, scientists discovered that this athlete carried a rare genetic variation in the EPO receptor gene. This variation increased the oxygen-carrying capacity of his red blood cells by nearly 50%, which gave him a clear advantage in endurance sports. Endurance sports focus on maintaining activity over extended periods, and they mostly rely on the aerobic energy system, where oxygen is used to produce energy. Well, here we see an extreme case, like an extreme example, where a single mutation has a dramatic impact. Usually, this is not the case. Usually, it's not just one variation, but many, which all together contribute to a trait, like athletic performance. Therefore, athletic performance and its related traits are considered complex traits. By studying a group of athletes, such as the skier we just saw on the left-hand side, which are referred to cases, and comparing them to controls, which can be either non-athletes or athletes from different disciplines, we can investigate the differences in the frequency of the genetic variances between the 2 groups. This study design is called a GWAS, or Genome-Wide Association Study, and is one example how to study sports genetics. This method can actually be used to study any trait, including athletic performance. And before I dive deeper into my research, I want you to keep 3 important things in mind. To effectively study sports genetics, we need a large sample size, a similar group of cases and a similar group of controls, and most importantly, the results must be replicated in independent studies. Now, Let's dive into my research, which is organized into several chapters. And I would like to present chapter 2 to 6 to you. Chapter 1 was my introductory chapter of my thesis, and chapter 7 was the discussion of my thesis. During my PhD, I had the opportunity to work with the Dutch cycling and the triathlon team. A triathlon is typically considered a classic endurance sport, isn't it? But here's something interesting to think about. What about the sprint distance, which lasts on average 8 to 9 minutes? Can we still consider this an endurance discipline? And this question perfectly captures the issue we discussed in the second chapter of my thesis, in which we critically examined sport discipline typologies. To start with, I would like to ask you a question. What exactly are endurance sports? Are 100-meter runners considered endurance athletes? What about marathon runners or swimmers or soccer player? Well, the truth is there is no standard definition. Several classification systems exist, but they are rarely used and they differ. And as a result, scientists often group athletes from different disciplines together as one group and label them either endurance or strength. However, as you can see in the pictures, the physiological characteristics between, let's say, a swimmer and a soccer player are quite different, and so is their genetic makeup. So we should not analyze them together as one group. Otherwise, the study results become difficult to replicate and almost impossible to compare with other research. The conclusion of this chapter is the following. We need to raise awareness of this issue, especially in sports genetics research. And there's a need for a universally accepted classification system to define sport disciplines, especially for sports genetics research. Well, to study sports genetics, we need a large sample size. And this is especially challenging for elite athletes, as there are not many by definition. But luckily, there's another option to solve the sample size problem. And this is what we have done in the third chapter of my thesis, by conducting a systematic review and meta-analysis. A systematic review summarizes existing studies. And our goal was to answer the question, which genetic variants are different in endurance athletes, defined as long-distance runners and cyclists, compared to non-athletes? And here is what we did. First, we searched for studies that had already researched this topic. Then we selected the relevant studies that could answer our research question. And in the end, we were able to analyze 43 studies. And the beauty of such a systematic review is that it allows us to reuse and combine the data of the previous studies, treating them as one large data set. In total, we were able to analyze data from almost 4,000 athletes and over 10,000 controls. And what did we find? Well, we found 5 genetic variants were statistically significantly different between endurance athletes and non-athletes. To conclude this chapter, the findings suggest that elite runners and cyclists indeed do have a unique genetic makeup when compared to controls. Now, I would like to take a brief excursus into the fourth chapter of my thesis. In this chapter, we conducted a SWOT analysis, which stands for strengths, weaknesses, opportunities, and threats, related to the field of sports genetics. We did this by performing an umbrella review. An umbrella review is a summary of summaries or summary of reviews. The method we used is quite similar to the one we saw in the previous chapter, but with a different analysis technique. This time, we used a thematic analysis, meaning we looked at similar themes across all reviews, and then we categorized the themes into either strengths, weaknesses, opportunities, or threats. And what did we find? Well, we found several strengths linked to sports genetics, like advantages in technology, but we also identified many weaknesses, such as a low study quality. We also found many opportunities for the future, such as precision exercise. And finally, the threats were linked to ethics, commercial testing, and gene doping. To conclude, this chapter highlights that sports genetics hold great potential to contribute to the performance and well-being of athletes. However, there is a need for advancements in methodological and ethical guidance to address the field's limitations and threats. In chapter 5 and 6, we focused on a more practical aspect of sports genetics. We had the idea to develop a polygenic risk score for endurance athletes, which are used in DNA tests. And in addition, we wanted to include only the relevant traits, those that really matter for endurance athletes. So the key question in this chapter was, what are the most important traits for endurance performance? And that's what we investigated in this chapter using a method called the Delphi technique. The Delphi technique works by surveying experts over several rounds to reach a consensus on a complex topic. And we involved coaches, athletes, and other endurance professionals as experts on 3 rounds of surveys. Well, at the start, we provided the experts a list with 120 potential traits that might influence endurance performance. Then, the experts rated each trait as either relevant or not relevant. And if at least 70% of the experts agreed that a trait was relevant, it moved on to the next round. After 3 rounds, 26 traits are considered relevant for endurance performance, which is also the conclusion of this chapter. Some examples of these traits include vitamin D deficiency, muscle fiber type, or coping with stress. In chapter 6, we examined the perceptions of athletes and coaches towards genetic testing in sports. Well, from the previous chapter, we know which traits are relevant for endurance performance. And in the next step, we developed the DNA test for endurance athletes, taking into account only the relevant traits, for which we had enough evidence to be included. Then, we offered the DNA test to a group of elite athletes. After analyzing their DNA, each athlete received their genetic test result in the form of an athletic report. Then we conducted interviews with the athletes and their coaches to gather their perceptions towards genetic testing in sports. In this slide, I provide some results from the athletes. The positive and negative perceptions are provided. On the positive, the positive perceptions are shown in green and the negative ones are shown in red. And what we can see is that most perceptions were positive ones. For instance, athletes felt that the DNA test had an added value for them and impacted the training. Negative perceptions, on the other hand, were related to the worry of misinterpretation or reaching wrong conclusions based on the DNA test results. To conclude, on the positive side, the study found that genetic testing offers great value for both athletes and coaches. However, there were also important concerns raised, which must be addressed in the future. To summarize, my PhD explored the role of genetics in sports with an emphasis on endurance athletes. The findings indicate that endurance athletes indeed possess a unique genetic makeup compared to non-athletes. This means that genetics can increase the susceptibility to endurance performance. However, it is important to acknowledge that endurance performance is a complex trait and is influenced by many factors and many interactions. Therefore, genetics cannot predict endurance performance. And sports genetics research is also not aimed at making such predictions. Looking forward, large-scale research is necessary to identify, replicate, and validate genetic variants that are linked to sport-specific traits. Thank you for your attention. I now give the word back to the Prorector.
Thank you very much for a clear presentation. It's now time to start with the opposition, and the opposition will be opened by Professor Prins, Professor of Clinical Epidemiology at Maastricht University.
Thank you, Mrs. Prorector. Dear candidate, first of all, congratulations with a beautiful thesis, beautiful book, and a lot of work. I think you read more articles than most people do during their PhD thesis. And I also want to compliment the promotion team because they did an excellent work in giving such a variety of techniques to study your goal. But I also have some questions because that's why I'm here. And my first questions are technical. I am quite specialized in meta-analysis and systematic reviews. You did both. You did a systematic review with meta-analysis in chapter 3. And if I then look at page 60, 61, and 60 to 63, usually when you do a systematic review and a meta-analysis, so you think you can add the data together and make a common estimate, then usually it's done based on sample size, with the lowest sample sizes down and the higher sample sizes up in the table, and you present a funnel plot. Mm-hmm. And my question is, why didn't you do that? Because I think if you look at that and did it, you might detect huge publication bias, especially that only very positive small studies are published.
Highly esteemed opponent, thank you for your compliment and thank you for this question. This is a very good question, and indeed, in chapter 3, we performed a systematic review and several meta-analyses. And if I understood your question correctly, you are referring to publication bias. Exactly. So what we did actually do well, there were several meta-analyses within this chapter, right? Within this article. What we did, we looked at the funnel plots, of course, for each meta-analysis, and we did not detect publication bias.
Okay, but for example, if I look at table at page 63, mm-hmm. The largest study has the lowest effect and the smallest study has the highest effect, which is usually a sign of publication bias. And similarly for, how do you say it, the table on 62, the smallest study have the highest effect. And yeah, so are you sure there's no publication bias, or are you just assuming it?
Thank you for this question. We assume there is no publication bias. We can actually never be 100% sure if there are publication bias present or not. Yeah, we analyzed the funnel plots and also by statistical analysis, mm-hmm. where we were able to analyze it. And on page 63, we are referring to a meta-analysis where only 3 or 2 studies are included. So the sample size is very low.
That's a limitation. Yeah.
This is a large limitation, unfortunately, in this meta-analysis, mm-hmm. Where we never can rule out publication bias for 100%.
Yeah. Okay, then another question. You put a prediction interval at down on all the tables you have, figures you call it, on the figures. Not for the last figure, because I think there's only 2 studies. But what struck me is that when you look, for example, at the upper figure at page 63, then the prediction interval is much larger than all of the individual intervals of the separate studies. And I looked at the method section how you calculated the prediction interval, but I couldn't find it. Can you explain?
Yes, thank you very much for your question. We actually applied the Hartung-Knopp adjustment for calculating the prediction interval, which has the effect that we have a larger confidence interval, or larger prediction interval around our effect size. And we did this when we included only 2 or 3 studies.
But you did also present a prediction interval for figure 3.2, page 60, where you have more than 10 studies.
Yes, thank you for this, for pointing this out. Indeed, we also here presented a prediction interval, and if I'm not mistaken, we used actually the mental Henschel method for the pooling, for pooling the effect size, and
which assumes a fixed effect, yeah.
With this assumes a fixed effect. Indeed, but it can also be applied to a random effects model.
Yeah.
So,
and probably that's more in the prediction interval.
Yeah.
Yeah. Because then you get less certainty.
Yeah.
Okay, then I have a totally different question. I still have time. When I was studying, and also during my training as internal medicine, I learned that the people from Eastern Africa, Ethiopia, Kenya, were so good in endurance sport because they had more white muscle, and the Western people had more red muscle. And that's what I always remembered. White muscle is for endurance, and red muscle is for sprint. How do your SNPs you studied relate to that red and white muscle difference?
Mm-hmm. Thank you for this question. If I'm not mistaken, you're referring to the muscle fiber type. Mm-hmm. And this is what we also found in our meta-analysis. A SNP, or a genetic variant, that has been investigated a lot in sports genetics research. This is the ACTN3 gene, which is researched, is one of the most researched genetic variants. And this is an interesting genetic variant in this protein, the ACTN3, in this gene, because if there are homozygous individuals which then actually lack the ACTN3 protein, which is responsible for fast muscle, like the fast contradiction of muscle fibers, the type 2 muscle fibers. And in contrast, when we are looking at the other alleles, these are responsible for, or these individuals have a higher proportion of slow twitch muscle fibers. And there we can see a difference in ethnicity. So you were referring to the different populations, and we can see a clear difference in their allele frequency between the ethnicities there, which is due to the genetic variations.
Yeah, I could go on with the discussion for long, but I think my time is almost over. And my next question would be on what, of course, you studied endurance because of the interest of your promoter, as I know. But there's also a lot of variation in agility with balls and so on. So I think there's much more to study in sport and genetics. So, and I give the microphone back to the pro-rector. Thank you for your answers.
Thank you, Professor Prins. The opposition will be continued by Professor Duking, Professor of Sports Science at the Technical University of Braunschweig in Germany.
Thank you for the word, Mrs. Pro-Rector. Dear candidate, congratulations to your thesis. I think you brought some light into the dark of sports genetics with quite different methodological approaches. So congratulations on that first. I come more from the practically oriented side of sports science. So, and your research showed basically that there are some genes which affect elite performance. And what do we do with these results now? So from a practically oriented perspective, if a coach approaches you now and says, I got 100 athletes, can you support me with your results on maybe which athlete to select? So what's the role of genetic testing in that perspective?
Highly esteemed opponent, thank you for your compliment, and thank you for this question. Well, it's a very good question and an important question. What is actually the practical implication of my research for athletes and also for coaches? So, first of all, it's important to acknowledge that sports genetics or sports-related traits are complex traits, as I said in my presentation, which is determined by genetics and also by other factors, environmental factors and interactions. So I think this is very important at the beginning before we actually start applying it in practice to know that it's not only determined by genetics. So we can only estimate a susceptibility to a certain trait, but we can never predict talent, because you were also referring to this aspect, can be maybe pick or identify talents by this genetic test. However, what can we actually do with this genetic test? So what we can do is to look at the genetic makeup of the different athletes we have and to tell them how they can individualize their training program based on their genetic makeup. Let me give you an example. For instance, we can look at the speed of lactate metabolism. There's a genetic predisposition for that, and then if we know that one athlete is a fast metabolizer, then this would mean, okay, he can start or she can start quite early again with a new repetition of high-intensity interval training, or let's say, looking at the bigger picture, the lactate threshold may go for like on a higher intensity for a longer period and needs less recovery time. So it can tell us actually personalized recovery times by looking at the genetic makeup, for instance. Yeah. Another very practical example would be looking at nutrition, for instance. So we could look at the metabolism of caffeine, the speed of caffeine, and also can I take caffeine at all actually, or should I better not take it? So caffeine it's known for its ergogenic effect in sports performance, and we can see by looking at the DNA, do I have a fast metabolism or a slow metabolism? And if I have a fast metabolism, okay, then I take it like half an hour before my competition or before the training. If I have a more slow competition, I take it 2 hours in advance or 1 hour in advance. So depending on that, we can adjust and really personalize our training strategies. And not only training, but also many factors, psychological factors, nutritional, recovery, training. Yeah, and it will give us a more clear picture how to train more personalized.
Mm-hmm. Wonderful. That would be fantastic if that would work. And are you aware in, for example, the show or use cases you mentioned for the metabolism or for the intensity of training, are you aware of any randomized controlled trials or studies which did use, let's say, genetic testing and then adjusted the training procedure? And did it then result in better outcomes compared if you would not do the genetic testing?
Thank you for this question. Indeed, there are several studies examining this. However, there are unfortunately not a lot yet, especially with this high-level athletes. But the studies that are out there indicate that there is a potential. And indeed, for example, Pickering in 2023, he investigated the speed capacity, like based on DNA test, and the effect of basically running fast. And indeed, he saw that in the group where the athletes had the gene for the ACTN3 gene, what I was referring before to, this group, which was lacking this gene, this resulted actually in gaining lower results or effects from this training, which is, I think, a very interesting finding. But in the future, of course, more research is necessary, because there are only, I think I can tell it on one hand, how many research has been done based on like personalized training approaches. So there's a lot to find out in the future, and I'm looking forward to it.
Mm-hmm. Wonderful. And in your studies, you identified 5 genes which make athletes elite, their unique genetic makeup, you call it. I'm interested in, are there genes elite athletes do not have? Let's say, are there genes which make me, or which impair me of becoming elite?
Again, a very interesting question, thank you. It depends. This really depends on the kind of sports you're looking at. Let's take again the ACTN3 example. It has been found that one group, endurance athletes, they lack the ACTN3 gene. On the other hand, strength athletes, power athletes, they do have the ACTN3 gene. So it's not necessarily to have it or not to have it. It really depends on what you're looking at, what is the trait and what is the sports you're looking at.
Mm-hmm. So it seems we need more studies on these topics, which is good. So we are not getting unemployed in the near future. And you mentioned that on your last slide that we need more larger scale studies, more multi-center studies maybe. And in your thesis, you also write that maybe other forms of technology, let's say wearables, artificial intelligence, could support us in that endeavors. How would you approach that, and how would you use these technologies in such future studies, and what are potential downsides of using such technologies?
Thank you for this question. Again, a very interesting topic, and indeed in the future there's many more to find out and to investigate, and hopefully we will not get unemployed. So by integrating, for example, variables or other databases, other sort of data, we could create, this would be a very nice thing to do in the future, actually, in my opinion at least, that we have one large database integrating variable information like real-time responses of the athletes. Many athletes are already now wearing variables and collecting a lot of data, putting them all together in one large database, and together with the genetic side and other omics, for instance, also epigenetics, of course, the proteomics, metabolomics, etc., we can get the whole picture, like a holistic view of how it works. Genetics is only one part of it, and it would be very interesting to pursue this kind of large-scale research in the future, and by also integrating, for instance, AI, which is rising, we can find new solutions and new techniques to investigate the whole picture on what makes athletic performance unique.
Mm-hmm. Wonderful. I think my time is up, so I hand the word back to the prorector.
Thank you very much, Professor Duking. The opposition will be continued by Professor Glatz, Professor of Cardiac Metabolism at Maastricht University.
Yeah, thank you. Dear candidate, let me join the previous opponents in complimenting you and your supervisors on your PhD thesis. It is an extended piece of work, and you studied sport genetics from various viewpoints, and I think that the title, Critical Appraisal, is fully correct on that. And also, I would like to mention that I was happy to read that you put emphasis on the fact that the findings from your thesis should not be extrapolated beyond the context of the professional athletes. I think that is important, because that may be a risk. Yet, there are a few aspects I would like to discuss with you. And I would also start with the topic that the previous opponent already mentioned, that is the 5 polymorphisms, the genetic variants that you identified in chapter 3. and that are significantly found. But in the first chapter, you already say that by 2023 there were 250 of these polymorphisms already identified, and then in your studies you went from 42, you went down, so now we only have 5. So how come that's a marked difference? Is that related to the poor quality of the various studies, or can you shed light on that?
Highly esteemed opponent, thank you for your nice words and for this insightful question. Well, indeed, by the end of 2023, there were more than 250 SNPs already identified. However, as you said actually correctly, these studies are not of high quality, mostly it's the case, unfortunately. This is mostly due to the low sample size. And these study results are not always replicated. And in our chapter in the meta-analysis, first of all, we focused on only endurance, which were defined by long-distance runners and cyclists, and the 250 SNPs in the beginning, they are referring to any kind of sports-related traits. So we have the first difference, why we have a smaller amount of SNPs. And we also focused on high-level athletes.
Okay.
So we had strict in and exclusion criteria, actually, and if we were not sure, we had to exclude this article, and we had to exclude many articles because the lack of definition of athletes. Like, what is the athlete status? Is it national, international? Is it amateur athlete? And also the sports investigated in these studies.
I see. So if we have better tools and a better design of the studies, we may expect more to find. Related to that, it is striking that these 5 polymorphisms are related to physical muscle performance, whereas I would think that motivation and coping with stress, as you already also mentioned, is also an important aspect. So how come you did not find genes related to those aspects?
Thank you. This is a very interesting question again, of course. I think or in my opinion, we would need a larger sample size to find those kind of traits, like psychological traits, because there are studies out there that have found many genetic variations linked to psychological traits, like coping with stress. This also has been included in the total genotype score we actually developed. But
so they did not reach significance yet.
Exactly. Looking at the small amount of participants we had only with running and cycling, we were not able to find SNPs significantly related to psychological traits.
Okay, that may explain it. Then I have some methodological issues. Of course, you have an entire chapter devoted to the better classification of the sports. But an important other aspect is the genetic testing itself. And that refers to how precise can we measure a DNA sequencing or SNPs. And I did not read about that. Is that, of course, a polymorphism may be missed for reasons of low quality of laboratory analysis. So is that an aspect that was studied, that was described in the studies that you investigated, or how do you read that?
Thank you very much for this question. So if I'm not mistaken, you're referring to the validity and clinical utility, actually, of the genetic test?
Yeah, and the precision of doing a whole exome sequencing or a SNP assessment. Yeah. I mean, if there is a few % mistake, then you may not identify a certain polymorphism, whereas it may be there just for laboratory standard reasons.
Yes. Okay. Yes. So, yeah, we of course considered this point, but we did not analyze the data ourselves. So this was done by third company, and the validity of this clinical test itself, like if they can correctly identify the genetic variant in it, was not in our hands, unfortunately. So we had only the results they delivered us, and I think in the future, and now with the new sequencing techniques, it gets better and better. And you were also referring to the whole genome sequencing. So this is coming more and more, and we have more valid and more accurate results.
Well, thank you. I think this is important. I mean, it makes a difference if it is a few tenths of percent or 5% differences, and so it's something to take into mind. Then, finally, I would like to go to a proposition that is not numbered and it appears on a page that is not numbered. So I would like one of your opponents, one of your paronyms, to read the proposition on the very, very end of your thesis, on page 320.
Winning isn't everything, it's the only thing.
Yeah, thank you. So I was surprised to read this statement because my thought was always as, well, you are, according to the Olympic thought, taking part, being a participant is much more important than winning. So is your opinion different?
Thank you for reading until the end of my thesis, first of all, and thank you for this question. But this is a very personal statement, to be honest. In looking at sports, I am the personality who wants to win. And I think it's also related to my topic here, because to reach the top, to have the Olympic gold medal, you most likely need a certain genetic makeup to really reach the top. Yeah. And it's not about having the second place, you want to have the gold medal. So it's a very personal statement, and it's also related to my previous soccer
thank you.
Playing. Yeah.
It shows your ambition and your motivation. Thank you very much for your for these answers, and I give the word back to the prorector.
Thank you very much, Professor Glatz. The opposition will be continued by Dr. Plasky, Associate Professor of Human Energy Metabolism at Maastricht University.
Thank you very much. Dear candidate, also from me, congratulations with a very beautiful thesis that you have written. It's not entirely my cup of tea because I don't know that much about genetics, and I'm also maybe a sports physiologist, but not so much into the techniques that you have used. So I hope you can help me out with some questions that I still have. Yesterday I was also sitting here, well actually where Professor Duking was sitting, but we were talking about precision nutrition. And in that thesis, precision nutrition was used to come to a better approach to improve health in individuals. But they used specific phenotyping more than genotyping. So they phenotyped certain populations, and based on that they decided we should treat them this way and we should treat them that way. So phenotyping, I mean, after genotyping, you need your genes to become to a certain phenotype, but we also know now that genes are maybe written in stone, but the expression of the genes are not. So they can vary with the environment, with our behavior. What is your view on that? Because we can also do very extensive phenotyping in sports performance. How much is the added value of the genotyping, which is, as I understand, very costly from your thesis?
Esteemed opponent, thank you for your kind words. Thank you for this question. So phenotyping, in my opinion, phenotyping is very important. This also refers to the first chapter we actually, or the second chapter, the classification of typologies chapter, where we highlight how important it is to precisely phenotype, because otherwise you might not be able to actually have valid results from your genetic, or based on your genetics, like from your genotype. The genotype actually you're born with. So, of course, what is interesting as well is the epigenetics, which influences your genotype by, for instance, physical activity or nutrition, what you mentioned. So I think by starting with a precise phenotyping approach is very important. But of course, we also need the genotype, the genotyping. And by studying epigenetics altogether, we can find out like what the influence is on a specific phenotype, having it precisely defined.
So you said to start with phenotyping and add the genotyping, or the other way around maybe? Start with genotyping and add the phenotyping, I don't know. Would your measurements, I presume your outcomes would not change with methylation or demethylation, right? You are studying polymorphisms that are there or not. So also relating to Professor Glatz's question, if you do your test today on an athlete and you do it again next week and maybe the week after, would you get the exact same polymorphism showing up? Do you know that, if there would be, or how much variability could there be in the outcomes?
Yes, no, it's the way what you said. You are born with the DNA and it will not change. So if you do another genetic test after a couple of weeks, it will give you the same result. However, the phenotype can, of course, be differently determined, or it can change. If you train a lot, you might be phenotyped differently than when you are untrained. Then you are in the control group. So this is what could change the phenotype in the end, and this is why it's so important to look or to precisely define the phenotype, so we have no misclassification over there.
Okay, and the technique is precise or reproducible enough to come up to the exact same result every single time, is what I understand from you.
For the DNA test, it is, yes.
Yeah.
For the genotyping,
Okay.
it is, yes.
So you say you should definitely keep adding the genotyping to your phenotyping. You stand by your field. That's excellent. You should. I'm very pleased to hear that. So maybe your field can help me out, because I'm much more working. I'm actually a health scientist, so I work with the general population, children, elderly, chronic disease, trying to study energy metabolism, physical activity to improve health. How much of you could I learn or apply in my field of research? Because then, of course, you had a very specific population, elite athletes, which makes it small, which is a disadvantage. But on the other hand, if I study the general population, it's huge, but there's going to be enormous variability, right? So how could we learn from, or how could I learn from your field to bring my own field a little bit further, maybe, than... Mm-hmm.
Thank you very much for this question. So in my research, I studied a very specific group, like athletes, high-level athletes, endurance athletes especially. So the question is, if I'm not mistaken, how could we actually apply this kind of research to a wider population and use it in public health?
Yeah, well, how could I get people more physically active, for example? Could I motivate them more? Could I improve compliance to interventions? That sort of...
That is something that I did not study in my research, but of course there are a lot of public health initiatives based on genetic testing. For instance, we also had in our department a project called Health Potential, which exactly looks at the general population and looking at specific genetic variants which could increase disease risks, but we could also look at traits such as physical activity in general or sedentary behavior. And there we have actually the opportunity to study large populations by using, for instance, the UK Biobank or other sources. So there we have a large sample size, and many genetic variants have already been identified and replicated and validated in order to find out genetic variants that are linked to physical activity. And then if we know these genetic variants, we could actually promote for the high-risk population activity programs or educational programs where we promote actively to foster a healthy society.
So make it more tailored and then hope that compliance will become better in the long time. Okay. Can I ask one last? No.
Very short,
Very
and
short.
a very short answer.
What would be your ideal next study?
My ideal next
Yes.
study?
I give you all the money you need.
Then I would come back to the issue I discussed with Professor Duking, with the big data and having a variety of sources, variables, omic data, genetic data, store it somewhere in a cloud. Of course, we have to make sure it's ethical and data is secured, etc. And then we could precisely phenotype based on, for instance, variable data, and look at the genetics of a certain population, athletes, certain positions maybe, and find out if there is a difference between the genetic makeup. Yeah.
Okay. Thank you very much for your answer.
Very
I
nice.
give the word back to the prorector.
Thank you very much, Dr. Plasky. We come to the last opponent. That's Dr. Forster, researcher in nutrition and movement sciences at the University of Saarland in Germany.
Thank you, Prorector. Dear candidate, also from my side, first of all, congratulations to a great thesis with a lot of mixed methods, and I personally really enjoyed reading the last chapter with the interview studies on the with the athletes and the coaches, which I thought was really interesting. So congratulations on that. I have a couple of questions nonetheless, and my first one also focuses on the meta-analysis, and I was wondering, and you touched a little bit on that in your discussion, you mentioned the original studies mainly included Caucasian males, and I was wondering what your thoughts are, how this influences your results, the impact on training, and what is happening with the females and with the non-Caucasians.
Esteemed opponent, thank you for your compliment and thank you for this question. I'm actually happy that you are asking because of course, I'm also a female, so unfortunately in sports genetics research, the majority of participants are males. Unfortunately, why unfortunately? Because we cannot extrapolate or generalize it to females. So we have to be careful what we are actually stating there. And unfortunately in our meta-analysis, the majority were male. We did test for interaction for sex, which was not significant, but what we also wanted to do, would have liked actually to stratify, although it was not significantly the interaction term, because then we could have actually seen, is there a difference between male and females regarding their genetic makeup and related to the performance outcome. What we actually see in other studies, that there is a difference between male and females, or at least in some studies which indicate that there is a difference in their genetic makeup. And you were also referring to the
Caucasian. Ethnicity?
Ethnicity.
Yeah.
Yes, thank you. Yeah, to the ethnicity. For ethnicity, it's of course the same. So we should also not analyze different populations together. So this is population stratification. What we also have done, so we looked in our meta-analysis for the different ethnicities, and what we saw, most of our participants were Caucasians, so we did analyze them together, but we also excluded the studies in a sensitivity analysis which were not Caucasian, and we still saw the same effects. So, yeah. And back to the gender or to the sex discussion, I think that in our findings we might not have found differences actually, because these were quite famous genes or well-investigated genes where other research also didn't find differences between male and female. But as I said before, there are genetic variants that are different, looking for instance at hormones or menstrual cycles, something like that. Yeah.
So still a lot to do.
A lot to do, yeah, and also more participants that should be females that also should participate in research.
Thank you. My second study goes a little bit in a different direction because it comes to bias, but I'm referring to the Delphi study you've done. And as you described your participants, 10 out of 18 participants in round 2 were academics. So PhDs in sports science and professors in sports science with, from at least what you describe, a focus more on physiology. So I was wondering if you think your consensus is biased towards physiology and if it would look different if more psychologists, for example, would have been included.
Thank you for this interesting question, and I'm very happy to engage in this discussion. So the positive thing about a Delphi study is that it can actually reach a wide group of participants, and that is also what we tried. We tried to reach international experts on the topic, and we had some pre-specified criteria which they should met. Of course, they should be experts with regard to endurance, but otherwise we did not have any strict inclusion criteria. So, yeah, it was chance that 10 of the 18 were high-level educational professors, and I think we should replicate actually the results, having a wider population or a wider panelists like experts, yeah, to see if there are differences and if our results were biased indeed. However, I still think that most of the key factors would most likely stay the same, independent of the expert panel.
Okay.
Yeah.
Would be interesting to replicate it though. My last question is, I think, goes beyond your thesis and is probably a little bit of a parental view. I would like to have your opinion on how important do you think actually genetics are for high performance sports or for sports in general in comparison to aspects like social status, access to training, support of family, encouragement of being active as a kid, so which happens much earlier. So what is your opinion on that?
Thank you. Well, I think, of course, that genetics is very important. Otherwise, I would most likely not have spent the last 6 years researching it. No, it's an interplay. I think it's equally important to have your genetic makeup and to have the environmental factors. Without the other, will not make it. You will not make it to the top. But looking at the like just for fun sports, then I think genetics is not that important, because with a lot of training, you can still, of course, you can still be a great runner or taking part in the Berlin Marathon and have a great time there, achieve a great time. So, but when we are reaching more and more the top level, like speaking about Olympics, then it comes, or this is at least what I think, genetics becomes more and more important. So it depends on the aim. And I think also without a certain genetic makeup, most likely will not reach the top.
Thank you very much, and I give the word back to the prorector.
If you have another question, you can ask it. Yeah, okay. If there is some time.
Okay. Maybe just extending on that, and again, probably more your personal opinion based on the interview study you've done, would you recommend genetic testing to athletes and coaches? And if yes, at which time of their career? Very early or late?
Well, sorry.
It's okay.
You can answer that question later on.
Later on.
Yeah, yeah, okay. Yeah, so right now the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return in this room.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. The line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Magdalena Schellenberg-Konopka. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Zegers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Magdalena Johanna Schellenberg-Konopka, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Citrius, alias Fortius, is the motto of the Olympic Games, an event where raw talent, genetics, and maximum training accumulate into performances of the highest level possible. These three Latin words mean swifter, higher, and stronger. And so are you, Magda. Swift, high, and strong, and you like winning. You always had an interest in sports. When you were young, you played soccer for Nuremberg on the highest level. And over the years, you became more interested in endurance. Now you're in marathons, triathlons, for which you need stamina and endurance. Even in your PhD in sport genetics, you took the emphasis on endurance sports, which I like, of course. And indeed, also in the PhD trajectory, it felt sometimes like a marathon and a triathlon, with ups and downs, long days, long years, suffering and overcoming obstacles. But you have done it all. And you have won. As we have heard, during your presentation and defense, during your years at Maastricht University, you conducted a massive amount of work. You calculated evidence-based polygenic scores for a large amount of sport phenotypes, systematic reviews, Delphi studies. You invited a whole new type of review, the SWOT review, and you did a qualitative study. You are an independent and smart thinker, not afraid of questioning the status quo. You even proposed that the sport scientist should actually use a new definition and not just use what's now called endurance sport. So you gathered the right people around you and wrote an article on that too. And you were right. Now imagine having a daughter like that, not afraid to critically appraise, which were the two words of your thesis, everything. I wish I could have seen that. Little Magda growing up. Imagine that you have a wife like that, Christina, tackling every obstacle like an endurance athlete and with a wish of focusing on winning. I only know you as a PhD student, and I can only come to one conclusion, and I'll try to say that in German. Liebe Eltern, liebe Christina, seien Sie stolz auf Ihre Tochter und Frau. Magda hat bemerkenswerte und echte wissenschaftliche Arbeit geleistet. Es ist keine Übertreibung zu sagen, dass sie das Feld der Sportgenetik vorangebracht hat. Wir allen stehen auf die Schultern unserer Vorgänger, die Wissenschaft voranzubringen. Jetzt werden neue Wissenschaftler auf Magda's shoulders stehen. Das ist eine große Sache. Magda, stay swift, Citrius, strong, Fortius, and I'm sure you will end up high, Altius. So on behalf of Billy Sperlich and Professor Geert Rietjens, it was a pleasure to work with you, and we will miss you.
Yeah, dear Dr. Schellenberg-Konopka, also on behalf of Maastricht University, I congratulate you with the degree you have acquired today. It must be a special day after a long period of hard work. Of course, I also like to congratulate your partner, family, friends, colleagues who supported you during this trajectory. I'd like to congratulate the supervisors here and online with this beautiful PhD defense. And I'd like to thank the committee here present, especially our guests from outside Maastricht, Professor Duking, Dr. Forster. Your contribution to this PhD ceremony is very much appreciated. Thank you for that. Coming back to your PhD defense, it was really, for us, a very enjoyable event. I hope you enjoyed it as well, as much as we did. But I can tell you that the committee was impressed by your broad knowledge of the field and the literature and various aspects that you covered in your thesis, which was quite exceptional, I think. Of course, there still needs to be a lot of done, many questions to be answered before this really comes into practice, but you gave some nice examples of what people already can do with the results of your work. So that's very promising. Yeah, I think we now have come to the end of this official PhD ceremony, and I close this academic session.